All right. Hello, everyone. I am Cindy. I'm a portfolio manager, disease control from the Bioworks division from BioFirst, formerly known as Biobest. I'm happy to welcome you all to this webinar on FytoSave, one of the products we are distributing in Finland. Today, the manufacturer will give you some more information about this product. They're Phytofend, a company from Belgium as well. And we have today Nicolas and Raphael with us, who will be giving the presentation. I just want to let you know that the chat is open. So during the presentation, if you have any questions, you can write your question in the chat. And then at the end of the webinar, we will have 15 to 20 minutes left where we will go over all the questions that have been left in the chat so that everyone gets an answer to their questions. All right, without further ado, I will give the mic to Raphael and Nicolas.
Okay, thank you, Cindy. So good afternoon, everybody. It's great to see such a large number of attendees to listen and learn about FytoSave. So thanks everybody for being here, and thanks to Biobest and Cindy in particular for organizing this webinar. And so I'm Raphael, co-founder and CEO of Phytofend, so exclusive producer of FytoSave. I'm going to present you the first slides about the company, and then I will give the floor to Nicolas for the technical part. So we can start. Nicolas. Okay, you can move. So very rapidly, just for your information, the first idea about the active substance is from 2000, and then we created the company in 2009. We filed some patents about the active substance and the formulated product. And then in 2015, we got the registration for FytoSave in Belgium, Netherlands, and so on. And then for us, it was a great achievement. We got a label extension of our product on grape, which is actually our biggest market over the world. And then we built our facilities to produce this active substance and formulated product. You can stay. Yeah, thanks. So this is the picture of our nice building with the operational part, the operational department, let's say, with the production area of the active, the formulation area, and then the repacking line on the back, yes. And then at the first floor, we have a very nice research center. So basically our lab, where we are able to perform bioassay in presence or absence of disease on plants, thanks to grow chambers. To increase, let's say, our knowledge and our data package on the active substance COS-OGA. I will come later on this active substance, and to investigate a new molecule for the future. So as you can see, we are able to grow tomato, basil, flax, but also more exotic plants like banana, maybe less interesting for today. Then, okay, this is our mapping, and you can see that we are mainly selling to in Europe and in the Scandinavian country through Biobest. So it's important to really understand the positioning of the product FytoSave. It's nice to capture the mode of action of the active substance. And for that, it's always nice to come back to the natural interaction that occur in nature between a plant cell, this is the schematic view of a plant cell, and the microorganism. So as you know better than I, your crop may be in contact, may face bacteria, fungi, but also oomycete, like some downy mildew. And those microbes release non-self molecules. So molecules that are non-self for the plant. And the plant has developed the capacity to detect those non-self molecules thanks to receptor or located in the membrane. On the other side, bacteria, fungi, and so on release in the environment some enzymes that are able to cut down and destroy the plant cell wall. And more specifically, one molecule that you certainly know in the cell wall, pectin. Pectin is degraded by pathogen because the pathogen needs to feed, to develop onto the plant. And the degradation of pectin leads to self molecules, so molecules that come from the degradation of the plant. And the plant has the capacity to detect also that it is being degraded by a pathogen. So the plant has really the capacity to detect, oh, pay attention, I'm facing a pathogen, but also my cell wall is being degraded. And this leads to a signal of danger. Then this is the principle. Now let's come back to the active substance, COS-OGA. This active substance is made of two sub-components, COS on one side, OGA on the other side. COS is an acronym for a long term, which is keto oligosaccharides. Basically, it is just a fragment of a molecule that is present in nature, which is called chitosan. And chitosan, you can find this molecule, for instance, in the exoskeleton of shrimps, but also of insects, in some mushroom, also in some extent. So we buy this chitosan, we cut down this chitosan in very small fraction that are called COS. In a similar way, we work with pectin. You know that pectin can be extracted from apple, or it can be also extracted from citrus peel. It's a byproduct of the agro industry. We cut down this pectin in small fragments that we call OGA for oligogalacturonan, but it is not important. The important is that we are producing those fraction COS and OGA that are recognized by plant receptors. And then what happens? In fact, COS mimic the presence of a pathogen. So it played the role of the self molecule if I refer to the previous slide. While OGA mimic the degradation of the plant cell wall, which lead and play the role of the self molecule. So based on those two kind of recognition, the plant initiate to mobilize a wide range of defense simply because it is put in a emergency situation because of this the fact that the plant believe, let's say, that a pathogen is present and it is being degraded by this pathogen. Okay, this is not really important. We continue to publish in scientific literature. This is also the signature of our let's say companies, that we are still very dedicated to research and the understanding of the mode of action. Please don't be afraid about those horrible names. Just in the past, we did a study on tomato. This is a crop which is interesting for some of you. We compare an untreated control with a plant treated with FytoSave, and we observe that many proteins involved in defense and stress response were overregulated, overexpressed in FytoSave treated plant. Okay, it is logical. This is the base. We are talking about a plant defense stimulator. It's logical that it increased the defense of the plant. But what was also interesting is that this product, FytoSave, was also able to increase the capacity of plants to make photosynthesis. And this explains an agronomical feature that we observed oftenly in the field or in the greenhouse about the green effect of our product following the application. So just for your information, the product is a cumulative product. So that means that you need to spray it on a regular basis. If you spray one time, okay, it's nice, but the signal, the level of the defense will not last for a long time. It's important to build the defense, to construct the defense with an interval between, let's say, 7 and 10 days. And this peroxidase is this is the level of peroxidase of it is a marker of the plant stimulation, and it's important to follow up this molecule in the plant because it is involved in the reinforcement of the plant cell wall, the defense proteins production. So the plant has the capacity to produce proteins that will be adverse to the microorganism, and then it creates an hypersensitive response. That means that if a spore of a pathogen, for instance, a powdery mildew, arrive onto the plant, it will have difficulty to spread out because there are some kind of very small necrosis, difficult to see with the eye, that limit the development of the pathogen. So you understand there that we will not, thanks to this product, we will not target the pathogen like a fungicide, a classical fungicide will do, but our target is not the microbe, but it is really the plant. So we are able, thanks to this product, to reinforce the plant and to make it much more resistant if and when the pathogen arrive. And now I give the floor to Nicolas to give you some example of efficacy and positioning.
Okay, so hi, I'm Nicolas. I'm a regulatory and technical manager at FytoSave, and I'm gonna try to explain to you a little bit more about FytoSave, and more importantly, what's probably more interesting you guys, the efficacy of the product in the field. So let's see what we can do with it. So, okay, what is FytoSave? Well, FytoSave will contain 12.5 gram per liter of Kosoga, the active substance Raphael just explained to you. So natural oligosaccharides. It has been recognized at EU level as a active substance, as a PPP active substance. So it's registered and all. It's a soluble concentrate, SL formulation, so really easy to handle, nothing specific about it. It's not toxic. It's not classified. It has no residue. So it's listed at EU level also as having no MRL, no PHI. Sometimes you got a PHI of 1 day if it has to dry up before going back in the field or stuff like that. But basically, there is no issue. It's approved for organic farming. So that's always handy, even though our biggest market for us is conventional, but yeah, we can go organic or full organic without any problem. It's a low risk, one of the few low risk active substance. No, there are more, but we were the first one at the beginning. It's a preventive product, so it's always better to be there before the disease enters in the field. And as Raphael said, it's a cumulative product, so you have to build up the defenses to reach a sufficient level. Okay, so what you have to know is that the activation response is very fast. So as Raphael explained, when you spray the product, the active substance is recognized by the plant, and the signal inside the plant is done quite quickly. It's a systemic reaction inside the plant, so the active substance won't enter the plant, but the signal will be systemic. And within 20 to 30 minutes, the plant is in emergency mode and is ready to start reacting. So it can rain after those 30 minutes. Doesn't matter. It will not be washed away. So yeah, the response is systemic, but to get it quite good, you need great coverage of your crop. Why is that? Is because, for example, on the guard cells, so near the stomata, the cells are more easily accessible to the active substance. So it's easier for the active substance to reach there, the membrane receptors. So it's always better to spray with larger volumes of water in enough pressure. So now that's the recommendation, but we've been working with water volumes way lower than that, and it still works. So no worry about that. Okay, so as said already, the effect of the application is cumulative and it has to be used preventively. The best case scenario is when you have had at least 2 to 3 applications before the entry of the disease in the field. Okay, so to get to that good level of defense, we would recommend to go 2 or 3 applications every 6 to 8 days at the beginning. So 2 or 3 to start it up fast. And after that, you can follow it up by a slower application rate, depending on the pressure of the disease. So every 10 to 14 days, you can continue and spray to maintain that good and high level of defense. Okay, so doing that, if you have a low or a medium disease pressure, FytoSave should, I insist on the conditional here, should be sufficient for on its own for the disease control. Okay, if the disease pressure goes higher than that and it's go too high to extremely high, it's necessary to complement it with other product. For example, in this case, contact fungicides. May they be organic or conventional? That's up to you, depending on how you're working. And basically, the best option, the best scenario is always to have like a complete program to defend and to protect your crop. You have to consider FytoSave not as a complete product by itself, but as an additional tool to put up in your toolbox. Okay, in terms of tank mixing, you can actually mix FytoSave with basically all or most of the crop protection products you're using. It mixes really well. It's very soluble. So remember, it's an oligosaccharide. So it's a complex sugar, and sugar in water usually mixes really well. So no worries there. It works beautifully with sulfurs, with coppers, hydroxide, oxychloride, sulfate, with potassium bicarbonate and other products more conventional, like all the one with cyfluphenamide, difenoconazole, mondipropamide, folpel, phosphatil, etc., etc. Yeah, we would recommend to always check it out in a small jar before to see if there's no incompatibility. But so far, from our side, we've noted incompatibility with tribasic copper, only the tribasic, not the other one. So that's not a really common copper formulation, let's say. But yeah, with that one, you get precipitation. And there are some active substances that we won't recommend to tank mix. You can use them in alternation. There's no problem with that. But tank mixing with phenexamate, for example, or spirotetramate, tebuconazole, it's less our recommendation to do so. But that's up to you. And in worst case scenario, just contact us and we can check it out. Okay, so let's look a little bit at the efficacy of the product in the field. That's probably why you were there. So the main target of the product is the plant defense induction. So as Raphael said, it won't target the pathogens directly. It will boost up the plant defenses against pathogens, mostly fungi. So let's look a little bit in cucurbits. We were here working in Spain. That was quite a long time ago. I was really at the beginning of the story of the product, and we were just comparing 2 dose rate of FytoSave vs the chemical reference at the time. Did 7 application and checked it out. Okay, so you have the untreated here, the lower dose, higher dose of FytoSave, and the chemical reference here. We are working here on incidence. The disease pressure is fairly high. You got an incidence of nearly 100% at the end of the trial. And you can see that the product keeps it under control quite good. But when you stop it after basically 2 weeks, it starts to go up again and then will meet up. But yeah, it's going to be about the same for the chemical too, but with a slight difference. For the severity, you will observe the same kind of curve. So fairly good control in severity, actually quite good, until you stop. And then you got 7 days without issues. And then after 14 days, when the effect of the product will disappear slowly. Okay, so we checked it out in a metabolomic process. So what is it? You spray the product, you let it sit for 24 hours for the product to really get into the plant and the plant to react. You grind the plant, you treat it a little bit and put it in a mass spectrometer with a chromatography, and you got the extraction and an analysis of the metabolites that you've got inside the plant. So really the reaction of the plant and what it did do when you sprayed, how it reacted inside. You can see that you got the control here. With just one application of FytoSave, we're still talking about cucurbits here. One application just one day before inoculation, you've got a slight difference. One application 7 days, you got a difference, but it's a little bit there. But then if you apply 3 times, you can really see a bigger difference between the control untreated and the plant treated with FytoSave. So the metabolites inside the plant are quite differently produced. And here, just for information, that's another of our product that will react quite differently, that will make the plant react quite differently. So for information. Okay. Another trials more recent that we did in collaboration with Biobest in Mexico. So we say, well, Mexico, yeah, but okay, cucurbits, if it grows, that will be kind of like a greenhouse in Finland, more or less. Okay, so you can also see the dose effect of the product and here in this case, organic reference, and we are in the same deal here, 8 application for repetition with an efficacy of the product on its own. Remember that it's a product that we would recommend in a program, but on its own, you got an intrinsic efficacy of 56 to 85% against powdery mildew on zucchini in this case. And thus, that's something that we always and would recommend if you're working with the product. And for example, you're working in organic, you probably spray or use sulfur. Well, you can work with a lower dose of sulfur and FytoSave before flowering, full dose of sulfur or triazole or whatever product you're using during the flowering, still with FytoSave to keep the defense level. And then you come back with the lower dose of sulfur and FytoSave, and you got, actually, that's what they do in Italy, for example. You've got a really clean crop, while the untreated is quite heavily damaged. So that's how we would recommend to work in cucurbits. Okay, so now let's have a little look at what's happening in tomatoes. I know that there are tomato growers amongst you. Okay, so same fairly old trial, greenhouse trial in the Netherlands in front of chemical reference. And on the severity of the powdery mildew, you can see that you have got about 50% severity, so fairly high disease pressure, and you've got a quite okay control by FytoSave on its own. And that's something that we have seen and observed repeatedly. Here again, another trial in Mexico, and you can observe the same. You got a dose effect of the protection. So for tomato, you're between 2 and 4 liters per hectare in the dose rate. So you're flirting with the reference here. So that's pretty good. What is interesting on tomato is that it also has a nice effect on Pseudomonas or bacteria, where you can see that 2, 3, or 4 liters, you got an efficacy as good as copper in the same kind of application scheming, let's say. So 5 application of FytoSave vs 5 application of copper, and you've got less severity on leaf or on fruit. Actually, you got pretty good control, considering that here you're 21 days after the last application and you still got fairly good control at 2, 3, or 4 liters per hectare. So quite interesting.
Okay, it was possible on tomato plant really to discriminate the impact of FytoSave against a control, an untreated control, in terms of metabolites. And then something really interesting. Can you hear me?
Mm-hmm. Yes.
Yeah, okay. Something really interesting is that in another trial realized in Italy, we observed that, okay, there was also some collateral effect, not only on powdery mildew, not only on Pseudomonas, but also on botrytis. This is a collateral effect. We are not talking about an anti-botrytis product, obviously, but it has really a side effect with a nice dose response. And you can see, in fact, it's not a dose response. The dose was the same, but the number of application was different. And the more you sprayed, the more you increase efficacy. So this is to summarize a little bit everything. This is the kind of positioning that we recommend. As long as there is no significant issue, you can still spray FytoSave. If something happens, you can also react with sulfur. Then you can come with sulfur or triazole depending on how you are working, and then to come back with FytoSave to manage the residue, eventually in mixture with a low dose of sulfur. At least this is really the way they love to spray. Also in Italy, when you can see really the difference between untreated there and FytoSave plus sulfur there. Okay, on pepper now. On pepper, you can see this is an official trials that has been carried out in Morocco by official by authorities, in fact, and you can see that FytoSave in green has been compared to an untreated control in red, but also the chemical reference in black. And you can see during the assessment that they did 3 applications, 1, 2, and 3 applications on 23 of October. And the disease was pretty well, we are talking about powder mildew, pretty well controlled, both in terms of incidence and severity. In baby leaves, I know that some of you are also interested in this crop. We observed, it was in Italy, nice efficacy against Bremia, so downy mildew on lettuce. You can see the evolution of the disease in red, in the untreated. In gray with another plant defense inducer, and then you can see the product. It is called here Eliseos, but it is not it is the same. It is a brand name equivalent to FytoSave, so physically it's the same product as FytoSave, and you can see that this FytoSave product is really competing with the chemical reference. This was in terms of incidence, but also in terms of severity, where the FytoSave is doing really a great job. Now, also on the high disease pressure and the greenhouse on lettuce. The same kind of result. It has been compared to laminarin, but also another chemical reference. You can see that this product with 4 application, 7-day interval, is doing nice job even after 14 days after the last application. Okay, then on Rocket, we are talking about the same group of crop, obviously, against yellow Peronospora parasitica. And again, we are talking about efficacy in the same statistical group than the chemical. And if you put FytoSave, so sorry, it is Eliseos, but as said, it is the same product. If you put FytoSave in mixture with copper products, so copper hydroxide and copper oxychloride, you can really get really nice result against this problematic disease. Okay, this is in terms of incidence, and now this is in terms of severity. Snow peas, okay, also a bit exotic, but as it is in the greenhouse, it can be also in some extent compared to your condition on snow peas, but it can be also extend to any peas or even beans. We can have a nice result with a dose effect, which is quite nicely observed here, but with 2 or 2.5 liter per hectare, you can get really nice result. Now we are talking about strawberry. We have quite a lot of trial on strawberry and berries in general. This first trial has been done in order to assess the dose effect of the product. You can see there that the dose effect is not really marked, let's say, but this is also a kind of trend for plant defense inducer. It's not like fungicide where, okay, the more you spray, the more you can have efficacy in terms of dose response. Sometimes with elicitors of plant defense inducer, you can see that clearly the interval between the application is much more important than the dose. So as I sometimes said, if you have money to spend for 5 liters, I prefer that you spray 5 times 1 liter than 1 time 5 liters, simply because the plant needs a kind of refresh, a kind of reminder, a little bit like a vaccine. Then, okay, this is another trial. This is the same trial, but sorry, but in terms of severity, and you can see that the dose effect again is not very impressive, but nevertheless, whatever the dose rate, you can see that the efficacy is very good compared to quite high, large amount of sulfur. And then maybe I will end with this slide to about strawberry, and I see that Nicolas is now back. He's now back, so I will give him the floor. But yes, in this trial in Italy, again, it was a powdery mildew trial, and you can see that the disease pressure of powdery mildew was quite problematic. FytoSave was doing a great job with 5 applications, 7 days interval. It has been also compared to a chemical, and also we tested FytoSave with sulfur, and you can see that this mixture is also doing a great job. I love this trial because at the end of this trial, the consultant asked us to see if we were interested to assess botrytis, and we say for sure, because there was some botrytis attack on fruits. And you can see that again, the evolution of the botrytis was quite important on in the untreated. The chemical was doing a kind of moderate action. It's logical because bipyramide is an anti-powdery mildew product, and it is not an anti-botrytis product. But FytoSave on its own was doing a good job and quite excellent in mixture with sulfur. So you can see that again, it is not an anti-botrytis product, but it has very nice and collateral effect, positive collateral effect on botrytis. We observe that on tomato, we observe that on botrytis, also on grape. So, okay, something is happening against botrytis, also in some extent. Now the following trial, maybe Nicolas, you can continue.
Yep. Okay. Okay. So we are back in Italy, still in greenhouse. So you can see again we did some trials more recently with dose effect of FytoSave at 1.5 to 3 liters comparison to bipyramide at 1 liter per hectare. We're working with 8 applications, 7 days interval, and the last assessments were made at 7 and 14 days after the last application. And you can see that you've got a nice dose effect, but already at 1.5, you got a fairly good control, and you're reducing it quite well at 2.5 liters per hectare, which is the dose rate registered in Finland. So that's quite handy. We are here talking about severity on the leaves. So you can see that a severity of 100% on the leaves, it's basically you've got a wide field. And here with FytoSave on its own at 2.5, you are under 5% incidence severity, sorry. So that's really good. If you're now looking at the fruit, so the severity on fruit still fairly high, over 50% severity, you also observe the dose effect, and you got something quite acceptable with just FytoSave. Remember that it's always better to work in a program with the product, so already on its own, it's not bad. Another trials where we did the same kind of application, but we got FytoSave at 8 or 5 application. So 10 days interval in that case, and in a mixture with sulfur, you can see that you're working quite good. And then in terms of statistical differences, you're getting something quite interesting around here. So quite interesting to know again for FytoSave, and you got the same kind of observation that you can do on fruits. So that's also quite a good player in fruits. Okay, another trials, this time in Morocco, again, like the first time, official trial, so we had no say in it. They were working as they wanted, although they were GEP, so they're still quite professional. What is interesting in this specific trial is the fact that the first application they did was with the disease already present in the field. So in terms of incidence, you were already over 20%, 15% more or less of severity. And with only 3 applications of the product, you can see that FytoSave is doing as good as the chemical reference, in this case, azoxystrobin, and gets a fairly good control, so over 97% on reduction on leaves, and a really good incidence reduction on the fruits. So quite interesting. So here we had our Dutch partner that was doing some trials to see the efficacy after the last application. So here you got the level of efficacy at the last application, and then 2 weeks after, you see where you stand. Fado being the commercial name of FytoSave in the Netherlands. You can see that the efficacy is quite good, and 2 weeks after is still quite there, over the economical threshold, which is really important for Dutch growers, while other products do their job, but maybe not with as good a remanence as FytoSave. So the boost of the plant is there for 2 weeks. That's what we observed also. After 2 weeks, you really see a decrease going on of the plant defenses. So you will say, okay, strawberry is good, but what about the other berries? Well, we have less data on that, but we know that, for example, on raspberry, so again, it's Mexico, but it's under greenhouse, so basically greenhouse considered about the same everywhere. You can see a nice dose effect. Not as good maybe as the chemical reference, but we are still talking about the product on its own, but still a fairly good control here on the severity on the leaves, so fairly high severity, over 55%. So an intrinsic value on its own between 50 to 70%. And on blueberries, you can also observe some quite interesting results. So here, this trial is a bit more complicated, but you can see that FytoSave on its own is doing something. And if you build it up in a program, you're getting even better results here. They started, for example, in that specific program with 1 application of sulfur, and then worked with FytoSave and a plant growth regulator, and had some really good control of the severity of the powdery mildew here. So quite interesting. So let's shift now on ornamentals. We'll go fairly fast because I see that we are getting to the end, but you can also see quite a nice dose effect of the product on on roses, in this case in Kenya, but again, greenhouses. For your information, with a high disease pressure, you're working on begonia, 3 liters per hectare. Only 3 applications, you would get about 80% efficacy. That was trials made by the Chamber of Agriculture in Germany. In 2018, they were working just to check it out with 1 liter per hectare, which is not enough, but they still had about 40% of efficacy of the product on its own. There was no pressure in viola and prunus crops, so they tried out the phytotoxicity and went up to 9 liters per hectare on flowers and observed absolutely nothing. So no burns, no phytotox at all. And in other crops, begonia, 3 liters per hectare, 3 application, they looked at the remnants of the efficacy. So 88% efficacy after 15 days, 71% still after 3 weeks, and over 50% 1 month after the last application. That's not something we would recommend, but it's always good to know that there is still something going on. Another trial here in Mexico, you can see again the dose effect of the protection. So intrinsic value 54 to 78% efficacy. And here a trial in the Netherlands where they observed some really good efficacy still on roses with either mixture of with a chemical or on its own or mixed with another biological product. So you got similar efficacy than the chemical reference. Okay, so it's basically, you can see it by the naked eyes also between the untreated and the treated plant with FytoSave. You can see that you got still a very good protection for powdery mildew. So why was Raphael talking about the green effect? Well, there is a biostimulant side effect of the product that will work and be observed in quite a lot of crops. So we know grape wine quite well. That's our biggest market. And what was done here, it's a trial in a dry year. So basically really few disease pressure. So the growers were asking themselves like, okay, why would I need to spray if there is no disease pressure? So basically they did their usual program on half of their parcel and the usual program plus FytoSave on the other half, and they observed in the production in liters of wine the difference, and they could observe like a yield increase that would actually pay for the product on its own. For leafy vegetables, what was observed by our Italian distributor is a shelf life increase. So as the product will slightly close make the plant slightly close its stomata, well, the shelf life will increase for cut leaves like rucola, lettuce, etc., and they will not lose weight nor water for some more days than the untreated. So that's quite interesting. And as you can spray really close to the harvest because there is no residue, that's always something handy also. Okay, yield increase in peas and beans also, you can really see the dose effect. And why not on cut roses? You could observe an increase in the shoot length of the roses, making them more easily sellable and at a better price. Still on leafy vegetable, you can observe a yield increase in lettuce, for example, here with pressure of Bremia lactuca, so downy mildew, and you can reach the same kind of yield than you would get with a chemical reference with the Eliseos or FytoSave at 2 liters per hectare. And in spinach, that's also something that we observed in a year when there was absolutely no disease pressure, we still had better yield of the spinach. So you would say, okay, that's not that much, between 16 and a little bit more of 17 tons of spinach. Well, yes, but it's per hectare. And if you put it in little packs of 300 grams of fresh leaf, like you will find in the supermarkets, well, it's still about 2,000 packs more per hectare. So that's good. For the information, you can really see it by the naked eye, also the untreated, treated here in hazelnut trees. So quite polyvalent. Okay, so I don't know if you still have the time to go through some testimonials, but basically here we got some growers in France, for example, okay, mostly aimed toward grapes, but they were happy because there was less downy mildew, and the FytoSave program worked as good as the chemical reference program. They could reduce the copper, which is really important that nowadays to reduce the copper, to preserve it and to be able to still use it later on. And so they were quite happy with that. No difference between the conventional protected plants. Another one in terms of quality, they have to reduce copper because it will antagonize the thiol precursor. For rosé wine, it's really important. And then they will be able, with FytoSave, they are able to reduce the copper to get to the quality objectives, still protecting their wines. So it reinforced defenses. Beginning of October, there were still green vines, which they are not used to see, and they have facilities to manage the residues. Okay, so they have yield increase, no need to treat when it rained. They went from 6 to 3.5 kilo per hectare of copper, and with really good control. They say that the product indeed has a cost, but with the yield increase and the tranquility of mind, because they don't have to, oh my God, it's raining, I have to treat again. Well, just for that, they say that the product pays for itself. So yeah, the other ones are about the same. So basically what is to remember here, it's that FytoSave is registered as a PPP. It's efficient against powdery mildew and some downy mildew, and it has additional positive effect against botrytis, as Raphael explained, biostimulant side effect, bacterial diseases, no residue, no MRL. PPP, just saw the question, the PPP is a plant protection product. Okay, so it has no risk of resistance buildup because it won't target the pathogen. It will target the plant and boost the plant. So it won't exert any pressure on the pathogen for it to mutate into a more resistant strain. It's not affected by UV and rain fastness. So within 20 to 30 minutes, the signal is in the plant, happens what happens, the plant will react. No phytotox, stable at room temperature, so no need to treat the bottle of FytoSave differently from the bottle of any other PPP, any other product. So yeah, can keep it there in your closet with the other phytosanitary product. No need to put it in the fridge or whatever. It's authorized in organic farming, a key tool for integrated plant management, and it's a patented technology. So that's about it. Sorry for having to rush, but as I was kicked out for a while, I don't know. I had to catch up.
Yeah, apologies for the little hiccup there, the technical hiccup. Don't know what happened exactly, but we got everything back on track, and that's the important thing. So if you have any more questions, please put them in the chat now, because I'll go over it. I've seen a few of them. So there was a question about about the combination with ozone water treatment.
Yeah, I saw this question, but honestly speaking, we do not have any experience with ozone treatment, and I'm not used at all about also this technology. I know that the registration of ozone at EU level is still pending from my knowledge, but a technical point of view, no, I cannot. We are not used. Nicolas, can you confirm? But I've never heard about treatment with ozone. But I'm curious, and I would like to know how does it work. So if you have any information about it, it could be useful. Maybe also to give you some piece of explanation, because if we know maybe the mode of action behind, we could maybe bring some information.
Yeah, no clue about ozone treatments.
Okay. Yeah, I've linked some of your papers from FytoFend in the chat, as there was a question if I could link some. So I've put just 3 just as examples, but there's many more. If you type in COS-OGA in Google Scholar, you will find a long list of papers. So definitely lots to read about for the active and the product.
Yeah, what is interesting in that is that we did publish some papers ourselves, but there are quite a lot of papers published by some other institute, some other university, from which we had basically no links or whatsoever with them, but they did publish and did their own studies. So it's kind of totally like independent from us. So that's always interesting to know also, because it's we are not just selling one stuff. It's just, okay, the active substance works, and yeah, it's not just us saying it.
No, yeah, important. There's also a question about the baby leaf results. I think this was the first efficacy results, if this was Lactuca or Brassicae.
It's mostly Lactuca, the trial we've got here, but we know from our Dutch distributor that on Brassicae it seems to be working quite as good. But from our side, we never tried it on Brassicae yet.
Okay. There's a question about the remark that you made that the product makes the plant partially close its stomata. Is there an increased risk of insufficient transpiration, for example, in high air humidity in the greenhouses?
No, but yeah. Sorry. Basically, what is important here is that when the plant is sprayed with FytoSave, it will react, but the plant will stay in control of all the reaction it has. So basically, it won't put herself in difficulty or in danger. It will just be ready to react to whichever pathogen will get, but it won't endanger itself or spend a lot of energy doing that. So it's just putting itself ready, but that's all. Like, you have to think about a runner, for example, a sprinter that is basically on the starting block, concentrated, but not doing anything yet. So not losing any control, not losing any energy, and not putting yourself in danger because it's not reacting yet.
And this closure is really, as Nicolas said, is really partial and a little bit like the efficacy, because it is linked. It explains also one part of the efficacy, and because this partial closure of the stomata limit also the penetration of some pathogen, like in grape, for instance, Plasmopara viticola, also the causal agent of the downy mildew, is entering in the plant through the stomata. So this is also one of the explanation behind the efficacy. And like the efficacy, it is also transitional. So it will not, this closure will not last for a long time. It would need to be repeated to keep the stomata again partially closed.
Okay, thank you. Yeah, PPP, I see you replied, Rafael's plant protection product. It's a product that's gone through the registration pathway in the EU. There's another question: what are the requirements for personal protection while applying the product in the greenhouse?
Nicolas?
Yeah, but it's like any personal protection you would use when applying something that is not classified. So basically, you don't need much, but it's always better to have like a coverall. But that just the basic ones are good enough, like for any application of a let's say safe product. So it's always better to be covered because as you will nebulize it, it will be really, really small. But as the product has absolutely no toxicity at all, you don't risk anything. But just put the classic basic one you have when you spray any biological or organic authorized product. Nothing specific for it, let's say.
Okay. And then we're back to the ozone. So the question is probably about the combination in the tank, because it is a strong oxidizer. So could that, being a strong oxidizer, could that decrease the efficacy of FytoSave?
I would say, first of all, I have to ask the question to our research team, and but then what we could do and what we do oftenly and on a routine base, we can make some specific tests just for for you, for some grower that are using this product, we can make some kind of test in our lab to see the compatibility, the physico-chemical compatibility in tank mix. You are right, but also why not a biological activity? So what is the impact of the ozone on the efficacy of our product sprayed on tomato plants? This is something that we can really do on demand, let's say, and very specific and dedicated to you.
Okay, that's great news. Thank you. I see no further questions. Does anyone else have any? Oh, yeah. Is spraying every 2 weeks in cucumbers together with, for example, Pre-Stop enough to see an effect? Is starting curatively together with a conventional product like Thalius and continuing with FytoSave from there a reasonable strategy? So I think that's two different questions. The first one is spraying every 2 weeks in cucumbers together with another biological product enough to see an effect?
Yeah, I would say for the first question, if yeah. It should be working, yeah, every 2 weeks. It would be better if at least to start it up, you could be able to spray it twice in a smaller in a shorter time frame. But after that, for the remaining of the crop, you can spray every 2 weeks. But if it's not possible, it will add something to Pre-Stop. Yeah, that's for sure. No, if you're working curatively, as you said, with a conventional product, you basically consider that the conventional product will do kind of a cleanup of the crop, and then you continue with FytoSave and start with FytoSave at the same time as the conventional. So conventional will cut it out and clean it up, and then FytoSave will boost the plant for the rest of the time. So that could be a valid strategy also, yeah, to be observed and followed up.
Yeah, it makes sense. It makes sense to start from something clean, thanks to a conventional application, and then when the main risk is over, then to manage the residue with FytoSave. It makes sense.
Okay. There's also a question about the strawberry results, where they can find the results after the meeting. So the meeting is being recorded, so the recording will be sent to everyone, so you will be able to rewatch if you want to. And I think available strawberry results, we can also put them together and pass them on to the technical advisors to share with whoever wants to see them on paper, that is. Okay.
Maybe something, if I may add one point, because we spoke about biostimulant effect, but let's be clear, this product FytoSave is really a plant protection product, registered as any conventional product with the same route, the same pathway, the same assessment way, and so on. But it's true that the frontier between biocontrol product like our products, so a kind of plant protection product, but from natural origin. So the frontier between this kind of biocontrol product and biostimulant that affect mainly the grow and the general health of the plant is sometimes very thin, but we are really speaking about a biocontrol product. And with this kind of technology, you can observe, and you have observed, that unlike a chemical that has a very specific action against one type of disease with a kind of 90 or 100% efficacy, here we are speaking about a product which affect positively the plant, make it resistant against different disease like powder mildew, pseudomonas, or botrytis, and so on, maybe with an efficacy which is intrinsically lower. We are very transparent and comfortable with that, but that can give you a solution, at least one part of the solution against many, several diseases. So it's also a kind of conclusion about this kind of technology. It's more kind of holistic approach on the contrary of the conventional product.
Okay. If there are no further questions, I think we can wrap up here. Thank you very much, Rafael and Nicolas, for giving this webinar. It was very interesting with some very interesting results. I think everyone has things to think about after the webinar. If there's any follow-up questions, please feel free to send them our way or to your technical advisors. They will make their way to us and we can still answer them afterwards. As I said, the recording will be sent out to everyone, so you can always rewatch. And thank you for everyone who attended the webinar. I hope it was informative and useful for everyone. Thanks again and have a nice day, everyone.
Thank you very much. Have a good day.
Bye.
Cheers.
Thank you. Bye.